Okay, I do hope you could see the PowerPoint presentation. Please do take note, mga classmates, that the copy of the PowerPoint presentation is already uploaded on your campus account. So, ito po yung first part, no? Akala ko kasi na-discuss ko na, kaya nag-go through ako sa use. So, this is your specimen collection and also your fungal culture. So we do have two PowerPoint presentation that are uploaded on your modules for mycology and for mycology. So yung fungal culture po and the specimen collection and fungal culture. So for mycology, it is very essential for us to collect appropriate specimen because this is considered to be the primary criterion for accurate diagnosis of mycotic infection. So sa lecture po natin, di ba, after the introduction to your mycology, the first topic would be your superficial mycosis or yung mga infection that somehow harbors our skin, our nails, saka ang ating mga hair. So very essential ang collection of appropriate specimen for the accurate diagnosis of mycotic infection. So please do take note, during your specimen collection for mycology, they need to be sterile. We need to practice aseptic technique. Another thing, swabs are not recommended for your filamentous fungi, and you need to collect adequate and appropriate specimen and submit and process them immediately. Most especially, yung mga specimen na gagamitin natin for Cryptococcus diagnosis. Okay, so these are some basic specimen that we can handle on the laboratory. But please do make sure, particularly sa handling, specimen handling, for your yeast culture, you need to have benchtop. Take note that all work on filamentous fungi, it must be carried in a laminar flow biosafety cabinet to reduce exposure to your fungal elements, because additional hazards of your airborne conidia, classified kasi sila as class 2 or they are classified to use class 2 biological safety cabinet. And the biosafety cabinet should be checked daily to see that none of the airflow inlets or outlets is blocked by supplies or incinerators or waste disposal containers. Okay, another, you could use Petri dish for handling, particularly for culture. Some labs do this for a better aeration and a large surface for colony isolation. And with the use of your Petri dishes, better handling and storing. However, it dehydrates yung ating mga culture. And hazardous, hazardous ang Petri dishes kasi madali siyang ma-open. Ideally, you could use your screw tops tubes. Yung mga screw top tubes natin, ginagamit po siya for culture. This is preferred because it offers less hazards compared to your Petri dishes, and this has a low or reduced dehydration of media. And syempre, yung atin pong mga chance of release of your airborne conidia is less likely with your tube media. So, if I may just add, ito po yung ating mga commonly specimen na natatanggap sa mga laboratories for diagnosis of fungal infection. So we have your hair. So yung hair, we need at least an epilate of 10 to 12 hairs, parang medyo marami. But yeah, that is the recommended. 10 to 12 hair sample. So pwede pong by clipping or plucking, no, from the base of the hair shaft. And then ilalagay po natin siya sa sterilized, no, or sterile container. Please do take note na wag po natin i-ref na yung ating mga sample. So after the hair, no, is put down on your sterile container, no, tapos na-receive na po natin sa laboratory, ilagay po natin sila sa sterile Petri dish and inoculate and incubate that for 22, no, to 30 degrees Celsius. Okay, another, we have your skin. Yung skin samples are usually scraped, no, from the outer edge of the surface lesion. Ideally, i-disinfect po muna natin yung site kung saan tayo mag-i-scrape ng 70% alcohol. And pagdating sa laboratory ng ating mga skin sample or skin scraping sample, we will subject that to KOH, no, wet mount, and it must be prepared to break tissue debris. For instance, kunyari yung skin sample from your scalp, particularly pag mga dandruff, no, you could brush that with sterile toothbrush or hairbrush, and the specimen is then placed in a dry, sterile paper envelope. Okay, another, mga nails natin. So, yung mga nails, it is submitted as scraping or cutting, no? Or the whole nail, kung natatanggal na, no, yung whole nail, you can subject that, no, or submit that to the laboratory. But first, syempre, we need to disinfect that with your 70% alcohol. And then, after receiving that on the laboratory, subject that to a KOH, no, wet mount for deeper scraping. Okay, another, we have your blood. No, yung blood, particularly from septicemic patient, no, they can harbor known pathogenic and opportunistic fungi. The most common being your Candida, no, species. Usually, this is collected if fungemia or the presence of fungi in the blood is suspected. So it can be collected, no, yung blood, no, and your brain heart infusion or your isolator tube, no. So this is considered to be a lysis, no, centrifugation system. And yung isolator tube natin, this is considered to be the most sensitive, no, method for the recovery of molds and dimorphic fungi. However, this method, no, has a high contamination rate kasi po yung lysis centrifugation system natin, it could somehow be contaminated, no, during the process. Pero the lysis centrifugation offers, no, better recovery of your histoplasma, no, capsulatum. And pwede din po kayo gumamit ng mga biphasic media such as your septic check, your Castaneda bottles, no, or yung mga automated systems po natin na ginagamit din sa bacteriology, your BACTEC, no, or back alert system for the recovery of fungi. Mainly, ngayon po yung mga ginagamit sa mga hospitals that are present, mga laboratories that are present, no, on the hospital. And you could also use, no, your blood culture bottles. No? You just need to incubate that at 30 degrees or 35 degrees Celsius if you are unsure of whether it is a yeast or mold. And nagko-collect po tayo na blood culture, no, in 2 to 3 sites. So basically, no, you could utilize that. Another, we have your body fluids. No, yung mga, for instance, yung mga respiratory, no, specimens natin, yung mga sputum, transtracheal aspirates, no, mga floral lavage, no. Basically, ito po yung madalas na isubmit na body fluids. For sputum, kailangan po diff cup, no, collection. The first morning, no, sputum is preferred, and then the sample is inoculated into your two plates. First plate, no, usually contain your cyclohexamide. The second plate is not containing, no, your cyclohexamide. So basically, no, those are sample. Pwede po natin i-collect for body fluids. Another, yung mga oro, no, pharyngeal candidiasis. This is easily diagnosed with your direct smear or mga culture. And yung mga nasal, no, sinus specimen natin, this is obtained, no, surgically, and then it can be plated, no, directly to media containing antimicrobial agent except for cyclohexamide, which can inhibit some fungi. So ito po yung mga specimen po natin na pwede po natin, no, gamitin for mycology. Ano pa ba? Banggitin ko lang din, no, yung mga eye or corneal scraping or eye discharge. Pwede din kasi yan, no, isubmit sa lab, particularly if you are somehow suspected na to have fungal keratitis. No? It is usually placed, no, directly onto slides and then inoculated, no, sa ating mga non-inhibitory medium. No? And say for instance, discharge siya, it should be concentrated by centrifugation and then inoculate to your subarodes, dextrose, agar. Yung CSF, no, pwede din po as your body fluids, but basically may rule tayo, di ba? Pag CSF yung sample. The first tube is for your chemistry and serology testing. The second tube for microbiology section. The third tube for hematology section. But for CSF samples that will be submitted on mycology lab, it needs to be centrifuged to obtain more concentrated, no, specimen. So you could use filtration, no? Pwede po natin gawin yan, or subject that, no, to India ink preparation and also your culture, no, inoculation. So those are some, no, method, no, with regards to specimens, no, that we do handle for mycology, no, and mycology laboratory. Okay. For your transport, no, nakalagay dito, in transporting sample, it can be folded and contained in an envelope. So the transport, for the specimen transport, no, specimen, no, to the laboratory should be transported at room temperature, preferably within 2 hours. Do not use, no, overnight or 24-hour sample. Pero may exemptions tayo doon, particularly for your hair, for your nail, and also for your skin. No? They can be used up to 72, no, hours at room temperature. Kasi po, yung mga pathogenic na fungi po natin, they grow, no, slowly, and any delay compromises quality, and it could also increases, no, the probability of isolating, no, the causative agent as a result of overgrowth or contaminants. Pero kung nag-expect ka naman ng delay, more than 2 hours, it should be stored at ref temperature or at 37, no, degrees Celsius. So ito po, no, yung sa skin natin. So these are some guides, no, with regards to your skin sample, no, collection. So sample must be scraped, no, from the outer edge of the surface lesion, disinfect, no, with 70% alcohol, and then subject that to a KOH mount to break the tissue debris, and this specimen is then placed in a dry or sterile paper. So these are some guides for your skin scrapings. So sa mga toe webs, saggy skin, it should be peeled down away, mga 10 to 15 millimeters square. So you will use your scalpel for this case. But please do make sure na hindi mo naman masusugatan yung inyong mga pasyente. Sa scalp, you could use your sterile tissue brush or a sterile toothbrush or your hairbrush. And sa hair, sabi ko nga kanina, 10, mas better, mas marami. Kasi parang mas magkonti, yung 46 hair. But you could use that kung medyo somehow hindi na ma-tolerate ng patient yung hair. Kasi ideally, kailangan pati yung hair growth kasama pag plinak mo yung hair. So epilate of 4 to 6 hair, pero mas better, mas marami, mga 10 to 12 hair some. And do not place your hair sample at ref temperature. You just need to place the hair sample on a sterile Petri dish and inoculate that and incubate that at a 22 to 30 degrees Celsius. With the use of your Woods lamp, it could identify your infected hair. This emits your ultraviolet light of wavelength greater than 365 nanometer, and it can be useful in identifying infected hair, such as this, yung mga nagflores. Okay, so another, yung ating mga nails. So it must be submitted as scrapings or cuttings of the whole nail. Pero kung natatanggal na naman na yung buong kuko, pwede naman yun, is-submit na. So disinfect with 70% alcohol and subject that to your KOH wet mount for deeper scrapings. But please do take note, yung mga gagamitin po nating instrumentation and equipment, it must be sterile. Okay, sa blood, we have mentioned this already. So blood culture tubes, you could use that. But again, this is not ideal for isolation of fungal sample. Mas better na yung mga isolator tubes natin, na yung mga biphasic media. But if wala po tayong choice, ito lang yung available na sample natin or way for us to culture, you could use culture media. But then again, this is not recommended. Yung mga blood culture tubes na. Okay, another, we have your bone marrow. Your bone marrow, kailangan po heparinize and inoculate at bedside. So you could, the use of your blood culture bottles is not recommended for your bone marrow. Okay, and Candida species, these are commonly recovered in your blood samples. For CSF, kailangan po is centrifuge to obtain more concentrated specimen. You could also perform filtration technique. And then after that, it could be subjected to your India ink or culture inoculation. So 1 drop of the concentrate is used for India ink preparation or latex agglutination for Cryptococcus and also for your Cryptococcus, and the remainder is inoculated on your media. If more than 5 ml is submitted, more than 5 ml na CSF ang sinubmit sa laboratory, it may be filtered through a membrane filter, and portion of the filter is placed on the media. So you could use media with antimicrobial agent, and it should not be needed because CSF is normally sterile. Okay, yung ating mga respiratory, no, sample. So many fungal infection have a primary focus on your lungs, no, particularly sa mga lower respiratory tract, no, secretion such as your sputum, your transtracheal aspirates, mga pleural lavage, no, these are commonly submitted. So as I have said earlier, no, first morning is preferred, no, particularly sa mga respiratory specimen natin. And then less viscous sample can be inoculated directly, particularly yung mga bronchio-alveolar lavage, no, yung mga bronchoscopy na specimen natin. But if the material is not too viscous, the specimen can be inoculated on the media with sterile pipette. And for very viscous, no, specimen naman, you could use your N-acetyl cysteine, no, a mucolytic, no, agent, no, para po ma-digest, no, yung ating mga viscous, no, specimen. Okay, urogenital and also your fecal, no, specimen. Particularly sa urine, po, pwedeng tumubo ang mga yeast. So you need to centrifuge the urine specimen and then inoculate them to the media. So this sediment that are used, no, to make smear for microscopic examination, and you just need to inoculate that to your media. Process as soon as possible to prevent the growth, no, of your bacteria, and the recommended processing time is within 2 hours after collection. If beyond 2 hours, you must need to store that at ref temperature, no, kung madidelay yung ating mga specimen. So first morning voided urine is the specimen preferred because it contains a high, no, concentration of the urine analytes, and we can also use your clean catch, no, suprapubic sample. Okay, if I may just add, you could also use mga prostatic, no, secretion. So you just need to empty the bladder and perform prostatic massage and inoculate, no, directly. Usually po, if the patient is somehow suspected to have infection or fungal infection, you could recover blastomycosis, coccidiomycosis, and also plasmosis. And tissues, no, pwede din yan, no? Pag tissue po ang isa-submit sa inyo, you should mince the tissue and transport that into a sterile gauze, no, with sterile NSS, no. Grinding of tissue is not recommended, no, kasi yung process na to might destroy the fungal elements, particularly if a zygomycete, no, is present. Okay, another, no, we have your fungal culture. So in general, your fungal, no, or your fungi do not share the broad range of nutritional, no, range of and environmental, no, needs that characterize bacteria. So relatively few types of standard media are needed for primary, no, isolation. So this include your suberodes, no, dextrose agar, your suberodes dextrose agar with antimicrobial agents. You could also use your potato dextrose agar or the slightly modified potato flakes, no, agar, and yung ating mga DHI, no, agar enriched with blood and antimicrobial agents. So we have your suberodes, no, dextrose agar. So this is considered to be a general purpose isolation medium with a pH of 5.6 that inhibits, no, bacterial growth. So this is most commonly, yung SDA po natin, ginagamit siya madalas no, sa hospital for the initial isolation of your pathogens no, and subprobes. And we also have your potato flake no, agar. Yung potato flake agar naman, ginagamit po siya no, to encourage no, growth of your reproductive no, structures. So these are some agar no, that you could use no, for the cultivation no, ng ating mga fungal. So basically SDA no, ang ating pong ginagamit. So these are details no, with regards to your subroads. Dextrose agar, your BHI, and also yung mga agar po natin with inhibitor at room temperature and at 37 degrees Celsius. So for the incubation of your fungal culture specimen, no, should be incubated up to a month. So medyo matagal. And examine no, periodically before reporting as negative. So most fungi grow optimally at 30 degrees Celsius. Yung mga yeast naman po natin, usually they grow within 1 to 3 days, while your histoplasma may require longer growth time. And the Petri dishes no, should be sealed with a paraffin or scotch tape to enhance humidity. Doon po sa mga fungi natin that do rapidly grow, no, mga 2 to 3 days pa lang, no, meron na silang cottony, no, aerial mycelia such as zygomycin. And for those who grow within 3 to 5 days, no, cultures with distinct border, no, yung mga white or pastel, white apron of new growth of periphery. No, these are your hyaline or your septile no, fungi. Pero may mga fungi po tayo na slow growing, mga 7 to 14 days. No, may mga fungi din tayo that do grow for 2 to 5 days. And they are considered to be dioecious no? Fungi. May mga 2 to 5 days to mature. So in that case, no, for the preservation of your cultures, you could use no, storage in water, freezing, use of your mineral oil, and also freezing no, or dyeing. No, for macroscopic examination no, of your culture, you just need to observe the colony no, description, no, kung woolly or cottony ba yung kanyang aerial mycelia, no, kung dark colony ba siya, no, kung delicate or cobweb ba yung itsura niya. No. So those are the things you may consider no, for your colony no, description. So basically, no, those are the way for us to collect no, specimen for fungal culture. So allow me again to share another PowerPoint presentation, particularly for fungal culture. Cultivation pala ng fungi. Okay, so we do have your cultivation of fungi, yung mga fungal culture natin. So earlier, we have mentioned your subroads, dextrose, no, agar as your primary no, culture media no, for your fungal cultivation. But we do have other no, agars such as your BHI, no, agar enriched with blood and antimicrobial. agents. So, aside from that, no, meron din tayong mga iba't iba pang mga culture media. No? So, we have your dermatophyte uh, test medium. This is basically used for your microsporum, your epidermophyton, mga trichophyton, sa mga hair, nails, or skin scraping and scaling ng scalp lesion. So this is used to isolate dermatophyte from your cutaneous lesion. Chloramphenicol and also gentamicin are incorporated to inhibit the growth of most bacteria, and your cyclohexamide inhibits saprophytic fungi. So in this case, this medium turns from yellow to red because of the formation of your alkaline metabolites when dermatophytes grow. So the pH indicator for your dermatophyte test medium is your phenol red, and usually the microscopic characteristic of your dermatophyte on your dermatophyte test medium can be seen from the colonies growing on this medium. Okay, another, we have your inhibitory mold agar. So pathogenic fungi exclusive of dermatophytes. We also have your potato plague, agar. So this encourage the growth of your reproductive structure. Yung ating mga mycocell or microbiotic, ginagamit po siya for selective media used to isolate pathogenic fungi, such as your dermatophytes, saka yung mga systemic fungi natin. It do contain your SDA, your cyclohexamide, and also your chloramphenicol. And we have other agars such as your yeast extract phosphate agar for pathogenic fungi, blastomyces dermatitidis, histoplasma capsulatum, and we have your chromogenic agar for your yeast and filamentous media. Okay, so another, we have your differential test medium. So we have your ascospore agar for the detection of your ascospore in your Saccharomyces species. We also have your cornmeal agar with twin AP and trifan blue. So it do contain 1% glucose, but this is not recommended for the production of your chlamydiospores. Basically po, ginagamit po siya to differentiate Trichophyton rubrum, which has a red pigment, and your Trichophyton mentagrophytes. And then we have your cottonseed conversion agar. So yung cottonseed naman, it do converts your mold phase of your blastomyces to the yeast phase. Another, we have your Japex agar. Ginagamit po siya for aspergillus. Then we have your Niger seed. So this is selective and differential enrichment medium designed for the isolation and preliminary identification of your Cryptococcus neoformans. They do have brown-black colonies due to the production of phenol oxidase that is considered to be a result in the deposition of melanin in the cell wall. So gumagamit po sila ng teasel or Gizotia seed. Same with your nitrate reduction method. So this is for the detection of your nitrate reduction in confirmation with your Cryptococcus. We also have your Christensen urea agar for Cryptococcus trichosporum rhodotorola. Tapos we have your potato dextrose agar for the pigment production of your Trichophyton rubrum, sporulation of dermatophyte, yeast fermentation broth, and yeast nitrogen base agar. Okay, so for culture media, basically po, we do use your SDA as the primary culture media. No, kasi ang SDA natin just is considered to be a general purpose isolation medium with a pH of 5.6 that could inhibit bacterial growth. Pero we could add mga antibiotics such as gentamicin and also your chloram, tenicol. But another for your tube media, it has the advantage of being safer to handle and less susceptible to drying. So ito po yung mga screw top tubes natin that are used for culture. Again, this is preferred since it offers less hazard and has a lower reduced dehydration of media. So the chance for the release of your airborne conidia is less likely with your tube media. And for the incubation requirement ng ating mga culture media, is to take note that this is only the requirement, particularly pag dimorphic, fungus 37 degrees Celsius, or you could also use 30 degrees Celsius. And this specimen should be incubated up to a month or 4 weeks and examined periodically before reporting as negative. So usually, most fungi grow optimally at 30 degrees Celsius. For macroscopic examination, you just need to observe yung colonies, kung flat ba siya, kung folded ba siya, kung rugose. Pag sinabing rugose, parang may radial or groups or diff furrows that radiate from the center like your spokes of a bicycle wheel. May mga cerviform tayo ng mga brain-like and mga verrucous, mga wart-like with rough knobs wrinkled or convoluted. So for macroscopic examination, there are texture classification. For instance, ito yung glabrous. This is considered to be a leathery or waxy with little, if any, aerial mycelium is present. Yung velvety naman, parang it do resembles plush or velvet fabric or suede. And it has a short aerial hypha and very few conidia. May mga yeast-like na texture tayo that somehow resembles your coagulase, negative staphylococci, mga bacteria-like. So, wala din siyang aerial mycelia. We also have yung mga cottony. So, they do develop when colonies produce long aerial hypha. Ano pa ba? Granular, yung mga parang powder, yung itsura nila. So basically, ganun po itsura or ng ating mga macroscopic examination of your fungi. And then we also have your slide culture. So basically, it shows inoculation on the agar plug. So we could use this to demonstrate natural morphology of the fungal structure and for encouraging the formation of your conidia in some poorly fruiting fungi. And the advantage of your slide culture is that it can be preserved indefinitely, and this is very useful when the slide culture from a known isolate is stored in a collection for future comparison against isolates awaiting identification. So basically, those are the things that I want to share to you for the topics fungal culture and also the specimen collection. So do you have any questions or concerns about the topic that we have presented this particular morning. Dalawa lang naman sila, medyo maiksi lang. In total, parang 22 slides lang. Specimen collection and also your fungal cultivation. So the copy of the PowerPoint presentation is uploaded and published on your Canvas account. I will share you the link of the recording of this meeting, and I will also provide transcripts for the particular topic that we have talked about. Paantay na lang po nung link later ng mga classmates. So that would be all for this particular meeting. If you do have questions or concerns, pwede naman po ako i-message. So yeah, I guess we could end this meeting. Paantayin na lang po ng mga links natin later for the transcripts and also for the recording of this meeting. So bye guys. Thank you for attending the synchronous session for mycology lab. Mag-ingat po kayo lagi, mag-aral ng mabuti. And sana po, lagi masarap ang inyong lab. Bye guys. Thank you.
Thank you po, sir.